Garin. Good morning, everyone. It's really a pleasure to introduce you, Clarisse Palma Silva. Clarisse Palma Silva is an associate professor in the Department of Plant Biology at the University of Campinas, Brazil, Unicamp, where she leads the Evolutionary Ecology and Plant Genomics Laboratory. She holds both a master and a doctoral degree in biological sciences from the Federal University of Rio Grande do Sul and a postdoctoral degree from the Federal University of Rio de Janeiro and the Royal Botanic Gardens, Kew. Clarisse's research focuses on the evolutionary biology of neotropical plant groups, particularly emphasizing Bromeliaceae. Her interdisciplinary approach, which integrates evolutionary biology, molecular ecology, and population genetics, sheds light on critical aspects such as genetic diversity, reproductive insulation mechanisms, and conservation genetics. Clarisse's research contributes valuable insights into a broader question surrounding monocot diversity and adaptation by using Bromeliaceae as a model group. I'm really, really happy to introduce you.
No.
Okay.
Gracias.
Okay.
Muchas gracias, Diego. Gracias. Well, good morning, buenos dias, guten Tag, bom dia. I'm a bit confused with languages now. And I would like to start thinking this is a very great honor to be here speaking for you, and especially because many of you are very good friends. And it's a little bit, I'm very happy that we have been here for 1 week and we have very good chance to talk and see your work. But at the same time, it's a little bit sad because it's almost finishing. Then we are already planning and thinking when we will see again. And then, well, but life is like that. It's really nice to be here anyway. And this week has been amazing. And I would like to sincerely thank the organizing committee of this amazing conference, and especially Melania, who is not here with us, resting for producing the next generation of, of researchers, and Adam that organized everything so perfectly. And we all agree that this conference has been very, very nice. And all of you for waking up so early in the end to be here is after all the nights, the drinking and talking is a kind of an effort, I understand. So today we will talk about one of the preferred monocot family, actually, Bromeliaceae. And I've been working on this family. I wrote here 24 years, then I freak out. Because now we are talking about 20 years easily, and then life is crazy because it's flying. But yeah, but then I've been working with that since 2000, and each project, each student, and each thing that each data amaze me because this family is really exciting and very fascinating model for studying adaptive genomics and speciation. So I would like to start acknowledging people and the institutions and the foundation. Maybe I will be a little bit late in my talk, then it's already done. I think the institutions, I didn't mention, but I'm a professor at Unicamp, but I'm 1 year sabbatical, many of you know already, at University of Vienna, working with Ovidium Palm. So I thank you very much because some of the results I present here is part of our collaboration there. And some of the other results are the PhD of Marilia. So you will see her, she's seated there, but you will see many pictures of her as well. And then it means that any questions, she's there to answer. It's fine. It will be easy. Paulo, also my PhD students, is part of this project and have been very helpful. And Luis Santos is a postdoc in the Ovidius laboratory, and he's Brazilian and have been very important for some of the results I will show you. Okay, so let's start. we know that biodiversity is not evenly distributed in the world. And one question that I have in my mind is always why there are so many species in neotropics. I think everybody think about that, right? It's not my question. It's a huge question for us, biologists. And to understand that, my approach is trying to understand how, what ecological and evolutionary processes have generated and contribute to maintain this hyperdiverse neotropical biota. Okay, so I'm very focused on the process, and then today this talk will be much more for the microevolutionary patterns. We have been seeing many macroevolutionary studies in this conference, but then we go for a deep, scale now. When we think about generation of species, we know that speciation, the process of generation species, is a continuum. Right? Is the continuum of evolution of reproductive isolation. And actually, I need a pointer. Tem computador? Okay. I forgot to get the pointer. Okay. So when we think about this continuum, in one hand, we can, in one part of the process, we can have single population, species population, and then the other one, strong separated and diverged population. But in the gray zone, how we call, we can find many different steps of the process, right? So in the end, what we know is that speciation continuum is the evolution, is the continual evolution of reproductive isolation. And this is what we will talk about today. Ooh. So with the idea of speciation continuum, in 2001, a researcher named Wu proposed the idea of genic view of speciation. So it is represented in this figure that he published later also in 2004. So the idea is that when we have the genome of two individuals of different populations and they are not reproductive isolated, most of the gene flow will pass freely among them. There are a lot of exchanging, but sometimes one or few loci are locally adapted to different in environment between these two populations, and then maybe these small differences are not exchanged because they are not fitted in these differences in different environment. But along the process, we see that this small divergence are increasing by linkage disequilibrium, right? And then the region that are not exchanged anymore are increasing, and then we still have gene flow in some parts of the genome, but some parts of the genome is not exchanged anymore. And this are increasing until a certain point that we will have a total reproductive isolation, right? Now we assume that our species doesn't reproduce, exchange genes with any other species, but we know that the genomes have been here for many years until our species now is alone and completely reproductively isolated from any other. Okay, so this idea of genic view of speciation also is important for the idea of speciation islands. These will be the islands, right? The genes we call genes of speciation. And then also the idea of portal genomes, because these will be the portals where we can exchange genes, okay? All right, so with this idea in mind, 20 years ago, this is where I'm thinking that everything is 20 now. 20 years ago, in the publication of this very important book for the studying the speciation, Quine and Hoare proposed to modify the biological species concept, including the idea that this biological species concept, initially proposed by Meyer in the 40s, could be accepting that species are a group of natural population that interbreed, but are not necessarily completely isolated from other groups. And with that, we assume, we know now that speciation can occur with gene flow. Okay, so this is an important point in the research of speciation when we accept that we might have some gene flow during the process. When we think about gene flow among species or during the process of speciation, we are actually talking about hybridization, right? And we know now also that hybridization is overspread, is part of the process, is overspread in the tree of life. So animals, plants, fungi, and many other organisms, we can see this occurring. So it's well accepted today. And in 2005, Jim Mallet proposed that like 30%, 10 from 30 will be, plants or animals will hybridize. I'm pretty sure it's a much larger number of that. But anyway, just to illustrate that this is a really large and widespread process. So it actually produced other two questions. Why is this process maintained in nature? And what are the outcomes of hybridization, actually, and how it's important for us studying speciation? So we have some outcomes, some potential evolutive genomic outcomes of hybridization. So here we have fishes, but then you can just think they are plants, okay? Orchids or bromeliads, any monocots you want. I thought about changing it, but I didn't have time, and then I thought it was not very fair with the Cyclidean authors that spend a lot of time doing that anyway. So, okay, we accept it to be the same thing with plants. So when two species hybridize, they maybe produce nothing. It could be, okay, no outcome at all. They could merge, actually. The genomes could merge. One species, the genome of one species can invade the other one, depending on the frequency of one species and the other, right? They can form hybrid zones like these. They could also exchange few genes with what we call introgression. So the whole genome is in one, is of that species, but just with few genes, right? And then they can also form new species, sometimes when allopolyploids, for instance. But the important here is that sometimes when we think about hybridization during the speciation process, we think a lot about this process, but we have all of the other processes occurring at the same time, at the same clade. In different times of the clade, they are not exclusive, right? It doesn't mean that if you have one situation, you don't have the other one. So, and this makes the process and the study really complicated. Actually, I'm here not to make things easy, just to make things worse. It's more philosophical than trying to help, actually. So yeah, so this is, the outcomes are very variable. And then we think, what is the whole of hybridization speciation? Of course, this picture here make us laugh, but then the idea is that hybrids are creepy, hybrids are maladaptive. This is a fake figure, right? Just to be sure, it's not what I'm talking about. And it's made by artificial intelligence. I get it in Google. So the idea is that when we think about hybridization in these maladaptive outcomes or freaky, of course, the hybrids could be maladapted, but it's also important that if the fitness of the hybrids are low, then this will reinforce the reproductive isolation and then will produce, help to the speciation process, right? Of course, they can be evolutionary dead ends. They can impede evolutionary biology and production of biodiversity because of loss of genetic adaptation, sometimes with genes that are well together, right? And then when the genomic shock of hybridization happen, maybe we can lose some important adaptations with different loci. And we can also have species fusion that will decrease the amount of diversity. So yeah, it could be not that important for generation biodiversity, but I would like to argue in the end to convince you that there are more positive outcomes than, I don't like the word negative actually, but it's part of the process, right? I really like this figure. It's not very new, but I always use it because it's It's not real. So in this possibility, in this species, we have a plant that might be not resistant to it's in the shade. It's not resistant to UV radiation, for instance. But this one is resistant to UV radiation. When they hybridize, and then very few loci of the resistance of radiation are integrated in this one, we will still have the species, this species that will say, okay, this is species A. Nobody will doubt of that. But then this species will be adapted, and we'll have this resistance for the installation, for instance, okay? This is just an idea how adaptive introgression occurs. And then the process actually might serve as a bridge for interspecific gene flow. This can increase diversity, right? Because this can also be important for exchange of these adaptive alleles between parental species. It's also a source of new the generation of novel genetic variation or evolutionary characteristics. And then it will promote greater resilience during environmental change. Right? So the process itself is, the whole process might promote this resilience. It can also promote genetic rescue if the population is genetically depleted and receive genes from someone else. And this will decrease the risk of population extinction. I think with that, you will believe that I really like hybridization, right? But it's very complicated, it's very variable, and I know it's a nightmare for most of you because I have seen this weekend, many talks along my career. So always when we think about hybrid, they are hybrids, we have a problem, this is a problem, this is not good, we are deleting the hybrids. The people are so happy that when they're sure that we don't have hybrids, because of course it's a complex process. So because of this, the variability of these outcomes, comprehending the full scope of hybridization, the evolutionary history of lineages are extremely important, but really challenging. And I like this study also with bromeliads, with the subtribe Tillandsioideae, where you can see the arrows here are the exchange, of course, within species of the same genus, okay, we accept that. But then many times among genus as well, right? So the process can be really, really complicated. But anyway, bromeliads are really nice, as we see the work I showed you before, for studying this process, because it's an adaptive radiation family. And many of the clades, many of the genus are also radiated, adaptively radiated genus. And it's a very nice way to study these, the process in the Neotropics. This species has almost 4,000 species. I'm pretty sure 3,600, the number described until today, it's correct, but I'm pretty sure there are much more species undescribed. And at least in Pitcairn they are. And 78 genera that diversify in less than 20 million years, which is really, really fast, okay? All right, with all of that, I would like to drink water. And to start showing this one example that we are working in my lab, and also part of the sabbatical project I'm developing in Vienna with Ovidio, and part of Marilla's PhD. And they are my cute Pitcairnias albiflus. I've been working with them like 15 years now. I did a postdoc in 2010 also with them, and then working with them since then. And then these pink flower are a hybrid. In the beginning, when I did a postdoc in 2011, we used microsatellites, and we saw a very low interspecific gene flow between populations of the same species. So this number here, theoretically, under the population genetics theory, we expect to be 1 or maybe 2, or minimum 1 individual per generation that will be expected to maintain the species cohesion, and we can see pretty low. And this is because this lovely species occur in this lovely place, which is Rio de Janeiro. And the rocks, these outcrops, are the inselbergs, they work like islands, right? So the populations are really geographically isolated. And I mean, it's not expected from population genetic view, but then it's expected for what we see in other bromeliads and other orchids and many other animals and plants that occur in these isolated environments. So very low gene flow between populations of the same species and interspecific gene flow between the species when they are in sympatry. We have two sympatric populations, Pão de Açúcar, Urca population, and then you can see that sometimes the gene flow between species are higher than between populations of the same species, okay? And then the other important thing is that they are asymmetrical. Pitcairn stamina donate more genes to albiflus and is more protected from introgression than Pitcairn albiflus, okay? So this is what I learned during my postdoc some years ago. I don't like to think about years anymore. Anyway, this work was done, still now actually, in collaboration with Professor Tânia Vente from Rio de Janeiro, when I did one of my postdocs as well. And then Tânia, during her PhD in the 90s, she studied many reproductive isolation barrier, prezygotic reproductive isolation barrier. During my postdoc, we also see post-zygotic reproductive barriers, genic nuclear incompatibilities, and also plastidial nuclear incompatibilities. Not talking too much about them now because we are running out of time. Anyway, so the most conspicuous, because when we see the flowers, the white and the red flower, the most conspicuous prezygotics, of course, is the flower morphology. They are pollinated differently. They have different pollinators. And the white one is nocturnal with scent flowers. They both produce nectar, and they are pollinator visited by different or some bees that might be the same for both of them. And then Pitcairn stamina is a diurnal. They don't produce scent. They do produce nectar and are mainly visited by butterflies, pollinated by butterflies, visited by hummingbirds. We are not completely sure. But anyway, we can see that's a very strong, very okay reproductive isolation barrier, right? So it could be okay, right? This is the reproductive isolation barrier between them. But this is why I say we are always going deeper and deeper and finding more and more about this process, because it's not that simple. Then when I returned to Brazil and start working in an institution, I keep doing these studies on mating system, the divergent of mating system, and trying to see how this affect and how important it is as a reproductive isolation barrier. And with a progeny array, we test the outcrossing rates in sympatric and allopatric populations, and we could see that Stamina in sympatry have much lower outcrossing rates, much higher selfing rates, and we know that selfing, when the pollen of themselves are completely protecting the stigma, it will protect from the interspecific gene flow, right? So this is an interesting reproductive barrier. And it happens why? Because they have also reduced ergogamy only in sympatric population, because in allopatric population, the outcrossing is high and the ergogamy is also high too. So the anthers doesn't touch the stigma. And this is what we can see here in this figure, right? So here's in allopatry, anthers doesn't touch the stigma, and here is in sympatry when anthers touch the stigma. So this study actually shows us that the evolution of flower characters displacement can promote the reinforcement of reproductive isolation, right? And with that, we start the revolution high throughput sequencing time, which is, I agree, a very, very, very exciting time to be a biologist at the moment. I always talk and feel that we are like in the 19th century when people start working with microscopes and see, oh my God, there are so many microorganisms. Then our life has changed a lot. So I'm pretty sure this technology is also like that. I'm super excited to be a biologist in this time. And then with this new technology, we have so many questions actually, but then 3 of them that I would like to discuss with you today are, has divergence between species occur with migration, evidencing a case of speciation with gene flow? Because everything we saw, okay, was nice, but then is it a real case of speciation with gene flow? And which genes or genomic region are possibly associated with reproductive isolation? And then we can see these isolation islands. And then if the hybrids promote any novel evolutionary adaptation? This is also interesting to understand in the process. Okay, so we did a return to Rio de Janeiro. I never show pictures of Rio de Janeiro in my talks, but this time I was so excited about how nice they are. And then I decided to show also, here is the city of Rio de Janeiro, and this is the city of Rio de Janeiro. As you can see, it's a very urbanized place inside of the Atlantic forest. It's 8 million inhabitants, I guess, right? I think it's a little bit smaller than, smaller than São Paulo, but still huge amount of people, huge amount of us there. This is Niterói. These 2 cities are connected by a bridge. And the plants we work, those 2 albiflus and stamina, are endemic to these rocks here. It's not very easy to see the rocks, and the pictures you will see better. So it's not that albiflus only occur here or stamina only here, right? They occur in both, but then we decide to get one allopatric population here, one allopatric population here, and 2 sympatric zones where we have the hybrids, the hybridization. Okay, so now I'm gonna show you some of the pictures. Here is Marília, which is there, collecting. And then it's also interesting to see that our collecting plants is not horizontal, but vertical. So ropes are our best friends to help us to reach there. And the climbers, we always need a climber. I'm getting old and I cannot climb these rocks anymore. I remember when I was in the beginning of my PhD, I went there with Thelma and Christian, and then I could walk all the stones here. I was like going and up, and then I never did again. It was like 20 years of frustration, actually. But anyway, these plants are not very easy to photograph, and then we decided to take a picture with Marília and Tami, is also a student in our lab, because there is this green background, so the plant is also not too big. And then if you try to make a picture of them in the nature, it's really difficult from outside, from a little bit longer. So this is the best option, actually. This is also the brave Marília climbing a rock in Chacrinha population. Here is the Pão de Açúcar, the Urca population, and here is the Copacabana. So, in the Chacrinha State Park. She needs to climb. You cannot see, but she's with ropes here as well. And here, well, it was too much for Marília. Then we got a climber to go there. It's in Pedra Bonita, close to the Gávea rock. And then Marília, in this time, she's just holding the rope for the guy doesn't go out and lose our sample, right? Very important. So here is the guy going down and getting our plants. And this last other population is Itacoatiara in Niterói, when we always have some helpers. This is a colleague from the Botanical Garden in Rio de Janeiro. Okay, so finish field work. It's really nice. We need to go back to the lab and start working on this data and so many plants. We collect all the plants and put in the greenhouses, because now we need a good DNA quality. So the greenhouses now we're getting full of these plants. But anyway, in this first study we have already published, is Marília Tavares, the first author. We test our first question, if there is a speciation in gene flow. Then we test for 5 different models with the red-seq data that we have in that time. The first one is migration, is isolation with no migration, kind of null hypothesis. We knew that it's probably not the case. And then we have isolation with a constant migration. Isolation is old migration. Maybe it happened in the past, but not now anymore. And then could be a secondary contact, because these rocks are so isolated that they could have been isolated, diverging allopatry, and then in a second contact, they hybridize. But then actually the most important model was, no doubt, that was the model that we model for the migration, more migration after the divergence of the species and less migration at the moment. So it supports that the speciation with gene flow is occurring, and then it's diminishing the amount of gene exchange as we expect with the evolution of reproductive isolation, right? Because if they are evolving, we will have less and less and less gene flow when they are in sympatry. Okay, so our question were easily respond with this very nice analysis. So we went to our more complicated, and I would say more excited also, question, which is which genes and which regions are involved in this reproductive isolation. In 2016, we published the transcriptomes. In that time, people liked to publish the reference transcriptomes, and then we did that with flowers and leaves. It's just a data set that people could use, and we used that. And flowers and leaves for both of the species. And then we also identified 4,000 positive selected transcripts using K-Cas ratio methodology that shows the positive selection. Okay, so we had HAD-seq, and we have the RNA-seq. It's a little bit up. And then with this RNA HAD-seq, that we had, we also evaluate the outliers with Specia-Adapt, and then we select the loci that have a huge divergence, more than 85%. So they are the loci that are really, really different, and we got the ones that are positively selected, and then we combined, we mapped them, the HAD in the transcriptome, and we got 17 genes that were extremely divergent with positive selection, that's showing that something might happen there. From the 17, for the 70, 12 of these genes were annotated with some functions. Then we can see and try to understand what they are, how they are involved in the reproductive isolation, since they are completely different and under positive selection. And 3 of them, the major pollen allergen, UPD, UDP, galactose, and inositol phosphorylceramide, and this I won't say, are involved. I tried many times and I forgot. No, I won't do all the genes. It's fine. It's there. It's like German, right? For this big word. So these 3 genes are actually reported in other plants to be involved in pollen germination, pollen tube growth, and pollen orientation, right? So this makes us thinking that maybe what are the whole of these genes and the pollen pistil interactions? They maybe are, we maybe have another reproductive isolation barrier here that we are not thinking before. And then, of course, these need to be tested, and this is why we also took the plants in our greenhouses. Then now we can test. It's very exciting because we are going from genes to the phenotype. And usually people go to the phenotype to genes. Anyway, the 4th gene, the jasmonic acid is synthetase, is also very known for pollen development during abiotic stress. Flower scent biosynthesis, if you remember, one species do produce scent and the other don't. And also involved in the defense response to pathogens and herbivores. Okay, so then we found those 4 genes that might be very important for our candidate genes, right? Important for the reproductive isolation. Okay, so then we collect so many individuals now in the second part for the PhD of Marilia. We resequence them with short reads and a coverage of 30x. And then we classify them with structure kind of method. And what we can see here first is that we have many poor individuals, really poor individuals, from allopatric and sympatric population. And then these individuals here only from a sympatric population are the hybrids. And then we see here what we call early generation hybrids, could be F1s, F2s, but very early. And then we see late generation hybrids that are the individuals introgressed toward albiflus. They will look much more like albiflus, but they are not poor albiflus. They are introgressed individuals. And they will also have introgressed individuals towards histamina in a much lower rate, right? Which was exactly what we saw with the microsatellites. When I did my postdoc, this asymmetric gene flow between species. Okay, so we collect some of these individuals. Marilia is still working on this data set. And then I went to Vienna to work with the RNA-seq. We collect the RNA-seq from leaves of some of these individuals, not all of them. They were 110, then here's like 60 individuals. And what we can see here, I separate by the color albiflus, histamine, and the hybrid. So this very colorful and complicated heat map will show differences in purple and resemblance in yellow. And what we can see here is that the two species are very well separated, so they do have very highly differentiated gene expressions. So it's also nice, I didn't mention that, because I want to mention here. They occur in a little bit different micro allopatry. They are in sympatry in the rock. You can walk and one here and the other there. But then albiflus work is found more in open rocks, and histamina is more in the shade, close to the vegetation and protected from installation somehow. And the hybrids are found everywhere. So it makes also sense, because then we can see that differential expression between two species. And the other things I would like to point you from you here is that the hybrids have this broad range. We find hybrids all over these differences in gene expression. So the hybrid is not one thing, right? It's not one outcome, doesn't express just one thing. And this is really important, easier to see in the SPCA, right? Here are the differential expression pattern for histamina, for albiflus, but then hybrids have this wide, broad range of differential expression. When we try to see, okay, what are the genes that are differentially expressed between the species? So we reached for the gene ontology terms. Gene ontology terms are like a, how can I say, abbreviation of the function of the genes. So their function will be put together, like defense response to other organisms that are the most overexpressed genes in Pitcairn albiflus. In red here we have the genes that overexpressed in albiflus, and blue overexpressed in histamina. So these also make us to pay attention, because we have this other gene in the other work, but then it's 37 gene overexpressed involving response difference and defense response genes. So it's something that we have never thought about, to be reproductive isolation between these two species. So then we also had the genome assembly chromosome scale. Marilia's working on that. I won't talk too much. She has a poster, 43, right? 43? No, 43 is yours. 42? 42? Anyway, it's outside if you want to know details about the assembly. And then she did that with PacBio Hi-Fi, which is a long read. We look from those 37 genes that overexpressed in albiflus, the defense genes. And one of them, what we have here, right? We have here one individual PacBio sequence. So each of this line is different reads of the same individual. Okay, they are not different individuals. They are only one individual. And these genes, one of the overexpressed in albiflus, but not expressed at all in histamina, doesn't assemble with the albiflus. So what we think here, we probably think, is that we have maybe inversion. And then this inversion might silence the gene in histamina. Okay, so this also open another question. Do you think I will have answers? But I just have questions, more and more questions. What are the whole of these structural variants in reproductive isolation? And this is also a part of the Marilla's PhD. So here we have a dot plot where we compare albiflus and stamina genomes. And it's very hard to see here. This is why I enlarge. Normally the collinearity is very okay, but then we see some spots, some places that we might have inversions. We might have, because of course we need to finish the assembly. Marilia will return to Brazil and do that directly. On Monday, right? Finish it, just a preliminary result. But the point is that we are very interested now on these structural variants, since we have those two long reads genome assembled. And we know that lower recombination will produce these speciation islands and increase the reproductive isolation among species, right? So we are going in this direction now, trying to understand how it will be. Maybe in our next monocot, I can show you more about that. So our third question, I will finish, 42 minutes. Is the hybridization promoting a novel evolutionary adaptation? And then if you notice, I don't know if you have noticed that before with our PCA, we have two individuals that have a transgressive gene expression. They are what we call transgressive hybrids. They express something completely different than the parents. And this, I like also this figure from Lauren Hisberg a long time ago. When we have these extreme phenotypes that can happen in hybrids, they might be important for adaptation to extreme habitats or conditions. And then we check which are the genes that are overexpressed in hybrids, only in hybrids. So we compare the hybrids with albiflus, hybrids with stamina. So we have 337 genes that overexpress in hybrids regarding stamina, 450 in hybrids regarding albiflus, but 17 genes are in both combinations. So hybrids always overexpressing these 17 genes, and 2 of the genes are involved in response defense. So definitely this plant is being attacked for something. We need to check what is it, right? And then we have another question, actually, that I have never taught, which were the whole of biotic interaction on the reproductive isolation. This is actually very exciting when we think about neotropics, because so many species always interacting to each other, and this could be something really, really important for the process, the general process. Okay. So I think I finished with my speculations and results and nice philosophical questions. And what I actually would like that you leave here today is thinking that hybridization can have a range of outcomes. They are not bad, they are actually part of the process, and we need to integrate them. Actually, bad are the phylogenies that don't integrate them. And this is the problem, actually. Speciation with gene flow is really, really probably more common than we previously predicted. We need to deal with that. We need to think about how to study a neotropical and tropical evolution with this in mind. We identified 2 other candidate genes and responses that might be associated to reproductive isolation, the pollen-pest2 interaction and defense response, that we will keep studying. And hybrids show transgressive differential expression, suggesting that they might express extreme phenotypes. Of course, we need to see if these extreme phenotypes have different fitness, if the fitness is positive or not, and what is the whole of these extreme phenotypes for the evolution of those groups. Okay, guys, thank you very much. I would like to ask you to go to the coffee and return in this room for the Bromevo. We are having our last section of this amazing and fascinating monocot family. And in the afternoon, afternoon, also in this first one, we will keep talking about this the importance of historical and recent hybridization in other monocot lineages that we like also, palms and Zingiberraceae, and general. Marcos will talk more generally, and Ovidio will be talking about our nice lenses. Okay, so thank you very much. Muito obrigada. Not sure if you have time for questions. I'm sorry for that.
No, I think yes, there is time for questions. Thank you very much, Clarisse, for a very nice presentation.
Thank you.
Now I like bromeliads and I like
now? Oh, now, Diego, 20 years of friendship. Well. And now you like bromeliads. Okay.
Okay, thank you very much, Clarisse, for this nice talk. It's for me, it's an honor to can see all the chapters of this long history. And
you were there in the first collection with Tanya when we started describing the hybrids, right?
I was the person who saw the first time.
Yeah, I remember that.
Pitcairn. Tanya, there's a rose Pitcairn in Pão de Açúcar. Okay, let's go there. And all this saga has started. And it's very nice to see how the questions were worked by your group. I am very happy with this. It's a very exciting way to see the problems we have in the evolution of the bromeliads. And so what I would like to ask you is, we have allopatric populations of both albiflos and stamina in some places, and the sympatric population in Pão de Açúcar. We can see a huge impact in the habitat loss in this hybrid zone. I would like to know from you, what do you think, in what extent the habitat loss for human impacts can trigger this process of hybridization? Because in Rize, for example, we, in a recent work from Camila Mello at the URGS, we have the same pattern in Rize, that we have the hybrids in the separate species, and we also did the morphometric analysis, and we have Bromeliaceae carinata with a Bromeliaceae simplex whole genome, and in the both. And it was a very impacted area. And so I would like to hear from you, what do you think about this? That if the loss, habitat loss by human impacts can trigger this process?
Yeah, I would say it's a very bad news, actually, that we have been impacted, this population. I saw some videos that concern me a lot. They're constructing things on this rock. I always thought that because the Chacrinha, for example, is a protected area, so it's more or less okay, we have the impact of the city, but then we know at least one rock will be there. We don't know what will happen with the rock that doesn't have the forest metrics around anymore in the long future, right? But Pão de Açúcar is a really concerning thing. And this actually is a very good question, because when we did this study, the Marília here, actually, if we have hybridization just because of our impact in these rocks, we will have probably this secondary contact model more important. And here I didn't mention, but the divergence were during Pliocene, and these hybridization were like in the beginning of Pleistocene. So we have hybridization since they diverge. So it's not a novel process that is happening just because we disturb the area and then they get contact. That could be the case for many groups. They really are in contact and exchanging genes since then. And this is why we think it's a very good suggestion that we have speciation of gene flow and probably sympatric speciation, in situ speciation. But of course, if they are disturbed and they are impacted, maybe it could change the pattern, could change the asymmetry, and then it's something that we can check, because we have this analysis from 2008, the samples from 2008, and then now 20 years ago, basically, it will be something nice to see, to compare the patterns. If they have changed in such a short time, then it can be our fault, actually, and it could be something added to the process also. Yeah, it's a little bit sad to think about the Pão de Açúcar being destroyed.
Okay, thank you so much. Unfortunately, we have little time, so we can go for a coffee and then later to the next session.
Okay, good.
And more questions.
Thank you, guys. Thank you very much.